All right. So, I don't know, welcome me or welcome you, everybody. If you welcome. So, everybody speaking Italian or? Perfect, perfect, no problem. I am Alberto Maria Cattaneo from the Department of Plant Protection Biology. Sorry, I was from the Department of Plant Protection Biology from other institution. I am from the Department of Biology and Biotechnology, a different department from the one you are studying. I am a colleague of Valentina. And this is a presentation on semiochemicals. I don't know if you have ever heard about them. It's a seminar of semiochemicals that entitled Semiochemicals Research Strengths for their identification and their use in integrated pest management. Since nowadays everything is also trendy towards the direction of sustainable agriculture, this is what I will show you something that I have done in the course of my activity at the Department of Plant Protection Biology for the Swedish University of Agriculture Science. So, just to introduce myself, I am a researcher and I focus on the chemosensory transduction of pest insects. In particular, I am a molecular ecologist on this point, and electrophysiologist. And actually, I'm working on fitness and also on symbiotic interactions of insects with bacteria and other microbes. Yeah, I am, let's say, particularly focused on plant pathogens, on insects that pest plants, or plants of horticulture, so pest insects of horticulture. And I spent a long time at the Swedish University of Agricultural Science. And I spent time also at the University of Lausanne, at the USDA, I was back to Italy in 2023, and I started teaching at the University of Pavia, where I have prepared also part of this presentation, but actually I am working here at the University of Pavia, not very far from home, because I am from Cornaredo del Pavia, Milan, so I was a bit late today because there were traffic, but in any case, it's fun being over anyway. So, I will use the Swedish pedagogic system and the Swedish pedagogic approach, let's say. So lessons have to be interactive. Don't hesitate to make questions, to stop me, and it will be fun, I hope so. And see me as a colleague, if you can help. And so the target is to go together. So we will see what's going on in terms of semiochemicals, integrated pest management strategies, but it's important that you also participate. If something is not clear because there are some things that maybe you didn't see before, please don't hesitate to interrupt me. And this presentation will not be shared, and you may not like it. So keep attention. But I have to say something. Last time this sentence was different. And so since I am experimenting as a researcher while I teach, let's try it, please. So maybe you will put more attention, it will be more interactive than expected. Okay? Any questions for now? All right, this is the total life. What I'm going to talk during these two boring lessons. In particular, I will first give you terminology. Maybe we could speak about plant pathogens, a little bit chemical ecology, semiochemicals. Anybody of you likes cooking, for example, or making pizza, these kind of things? When you do that, you need ingredients. So these are the ingredients. We learn these things a little bit, and then we proceed. I will talk about the ecology of horticultural pests and the use of semiochemicals to the pest control. There will be a specific example. And chemosensory receptors of horticultural pests. How many of you are in molecular sciences? None of you? Okay. Of course, so we can also face some very basic things in order to give you this information. And isolation of the chemosensory receptors of insects, and heterologous expression in transgenic Drosophila melanogaster, with like common human commensa, and heterologous expression in vitro, it means using specific cell lines. Plant pathogens are very wide. There are different kinds of plant pathogens. So I don't know how much you are into plant pathology in general. Okay, we're not going to talk today about viruses, bacteria, fungi, phytoplankton, and also other kind of pathogens. We will focus mostly on insects. That insects make the damage, larvae make the damage among insects. But there are many other plant pathogens which may be of interest. I consider that insects are important because they spread the pathogens, like viruses, they spread also other pathogens like bacterial or fungi pathogens. In any case, this is just to give you a slide of impact, but there are many kinds of plant pathogens, and more or less there may be semiochemicals for their control, but it depends. And one interesting thing that you may already notice is that several plant pathogens are wrongly called invertebrates, but invertebrates is a very bad word because it's everything which is not chordata. It is not precise, it's not scientific. It's better to call them protozoans. Later we will now see about that. Integrated pest management. What is another terminology? What you know what it is?
Yeah, combine various pest management systems to have a better result.
Exactly. So it's a sort of alternative approach to solve issues in agriculture, rather than by focusing on the use only of pesticides. There are other ways that deserve to be integrated and operated together. I mean, there is a framework directive in the European Union about integrated pest management. There is not a sort of a regulation, let's say. Even though it is getting more and more, I would say, integrated within the laws of the various countries. I'm talking about Europe because we are in Europe. And for example, we have this symbol of the National Quality Control System of Integrated Production in Italy. That business who produce their own products through integrated pest management approach can apply to have this law. But it's not like organic agriculture, which has a lot of laws on its back and a long history. It's something more recent, which is not particularly well legislated, but it exists and it is recommended. And there are at the local level some directives, also the regional level of the various regions of the various countries of Europe, to respect specific disciplines in order to be able to achieve an integrated production for the various crops that are produced. And I don't want to go along all of these details because it's not super important. The thing that you have to keep in mind is that it's a TOTEF protocol. So there are ways that is a sort of directive, is a sort of summary of this framework directive. There are ways that you can improve and make it better and better. So the target is to continuously learn from your mistakes in phase of applying the various strategies in order to improve the control without using chemicals and trying to find alternatives. And yes, as a mode of fact, the application of these principles, there are these 8 principles, means a continuous implementation. So this is what you can bring home to have an overall view of integrated pest management. Let's say rules, even if rules is too strong as a word. Chemical ecology. You know what chemical ecology is? Have you ever heard about it? It is at the base of integrated pest management. It's necessary. So there is this description that is very close also the subject area of your UPV department. Refers to the disciplines that studies the chemical signal and their ecological role in communication, defense and finding food and making partners among organisms. It focuses on the complex interaction related by chemicals within and between species. Okay? So it's the ecology, but seen at the chemical point of view, seeing chemicals like messages among organisms in order to interact each other. And you can imagine this thing not related only with insects, but any possible organism existing in a specific ecosystem, whatever the ecosystem is. It can be also the gut of a person. There is a chemical ecology there. There is a communication, the communication related by chemicals. And we make use of chemical ecology to integrate the pest manage different kinds of insect pests, for example, by adopting their chemical ecology. Semiochemicals.
I've heard that there are some kind of molecules that they can send messages between species, for example, like attraction of them or making them avoid to go there. And also it's in their nature, like pheromones or allelomones.
Perfect.
And we can use them in attracting or avoiding, for example.
Yes. And what the chemicals we were talking about in the previous slide. Any sort of chemical that is present in the environment and mediates communication among organisms can be considered like a semiochemical. Indeed, the definition is chemical signal that convey information to modify behavior by stimulating or inhibiting it. Because these chemicals in the interferences, so among the various organisms in the various interactions, bring messages. And these messages edit the behavior of the species which receive. They receive a message and they behave accordingly. So there are different types of semiochemicals, as you say. There are pheromones, for example, which are intraspecific. So within the same species, organisms of the same species communicate by these semiochemicals called pheromones. And allelochemicals, instead there are different types of allelochemicals. There are the allomones, which benefit the sender. For example, this fruit is attacked by this caterpillar. It produces an odor or a particular taste, which is repellent for the caterpillar. So in this way, the fruit can prevent the caterpillar feeding on it. It benefits the sender. But there are others benefiting instead the receiver, like the chiromones. The chiromones are scents that, for example, attract, in this case, a female to deposit on the fruit. It benefits the receiver. The receiver feels this scent and find its oviposition place and leaves eggs. And can go along with its fitness and survive because it perceives this scent. These are the chiromones. Then there are also the cinnamon, benefiting both the sender and the receiver. Classical example of the honeybee that feels the scent of a flower and goes towards the flower. It finds foods, but at the same time, the flower is impollinated. So both the sender and the receiver are in advantages if they, when there is this semiochemical communication. And there is the antimones that I wanted to list because according with the list of semiochemicals, they have also been mentioned, and there is a publication that is very unclear because this publication cites another paper from the same author and writes exactly the same thing. So it's not clear what antimones are, but according with what is written, produced by a sender and repellent for both the sender and the receiver. To me, it sounds like a very bad scent. Okay. I don't know what they are, but they are listed. And then there are instead the apneumons. The apneumons are the typical odors of putrescence, of dead corpse, and other kind of flesh or dead species in general, dead animals that are something dead, that emit an odor that attracts a specific insect, but at the same time it's parasitical. So emitted by another source, detrimental to a species living on it, but favorable to a third species. The third species, for example, here is this parasitoid fly the parasitoid wasp, sorry, that lay eggs into the fly that is attracted to these apneumons. Okay? Let's see semiochemicals in the environment. I use the example of moths because I worked a lot with a specific moth species, because the moths is the Morella, later we will see something about it, but moths are very well controlled by semiochemicals. There are a lot of strategies based on the control of various species of moth, pest and fruits. In general, in the environment, which kind of fodder you expect to have for in this hypothetical agro-environmental ecosystem? What do you think to see? What kind of fodder are there? What do you think? What kind of fodder may we find in this environment? Flower oil, otherwise? Fruit, leaf, and grass, exactly. So, for example, three odors, leaf odors, female odors. Female odors are important for the male to find the female for copulation. Microbes make odors. Microbes living on fruits, living on plants, and the flowers and fruits odors. Okay. So let's take them one by one. Let's take the various scenario. The male can find the female by searching for pheromones. Normally, the insect cross a specific plume, because pheromones, when they are released, they usually follow a sort of wing, a sort of wave along the air. And when they find the plume, they time by time they cross the plume continuously, and they are able to find it quickly. But also females can do the same by finding fruits, yeast, flower, or leaf odors. They are capable to trace what is emitting, and after copulation with males, to go in there to deposit, and are extremely important because they need to find these odors to guarantee to their larvae food source and survival. Okay. Also, larvae, which are the ones making the damage, have a very complex chemosensory systems to detect and find their hot foods or microbes, which are extremely important. Most of the odors are emitted by microbes living on fruits, not only fruits. Fruits are without microbes. They don't emit a bouquet of odors, which is very wide. So, and in the frame of control strategies, while time ago, decades ago, they were rather focused on the pheromones, attractive for males, we are going towards the direction to consider any sort of fodder in a broader region of chemical ecology, which can turn useful for different kind of techniques. As I said, in the last decades, this was the most successful a chemical ecology success story, the use of pheromones dispenser. This, in the coming couple of slides, will introduce you to the most common way that chemical ecology turned useful to target and limit pests, like, for example, moths. Have you ever heard about methane disruption? Okay. Insects communicate by chemicals. So they don't care how the female look, they care how it smells. And the way that they communicate by chemical can be resembled to the way that we communicate by sound and vibrations, by voice. So now you can hear me because I'm giving you a signal. And it is exactly the same for insects. Imagine an example that I like to take. Yourself being together, you and a friend of you, in a big gym with a very wide open space, and you are blind, you cannot see anything, but you can hear the voice of somebody calling you. But imagine that this person is calling you, but at the same time, around in the space, there are various speakers emitting exactly the same voice, with exactly the same frequency, with exactly the same background. And so you may easily find the wrong way to find your friend. You don't find it. You find, I don't know, a picker or something like that. This is exactly what happens. By placing around the plant's canopy different dispensers emitting exactly the same odor of the female, the male doesn't know where to go. It may most likely land on the pheromone dispenser. It may waste its time, which is precious. And it maybe can copulate, but rather than copulating 10 times, it copulates once. Or maybe it doesn't copulate at all. Maybe it gets afraid, okay? Maybe it gets lost. This is how mating disruption works, by pheromone dispensers, confusing males in order to don't let them find females. They waste their time. There is still an impact of the pest on the crop, but it becomes less. And this strategy can be integrated in the frame of integrated pest management with other strategies based on the use of chemicals, based on the use of parasitoids, based on traps that can also be based on semiochemicals. So, massive trapping of female, males, larvae, or the insect of the species by other specific for the species, the so-called attracted kill. Well, this is an example. One example is sufficient, and this is how it works. The use of semiochemical for mating disruption. Okay, we can go over. All right. So, in vitro, these are the approach and these are the effects. There are dispensers that are made of semiochemicals which come from synthesis. Still, it pollutes, synthesizing this compound. It pollutes less than using pesticides. And they have a specific recommendation on the number of these dispensers that can be placed on the canopy. They suggest, depending on the feature of the field, how many to put. And the intent is to release a large amount of sex pheromone, to permeate the air, to saturate the air, hypothetically. And it is important that this approach is coordinated because there are regulations from the consortium, for example, or from the region that recommend that all the producers of this specific crop use the semiochemical control strategy together independently from where they are located in order to extend, like an oil stain on all the territory, the use of semiochemicals. Why? What happens if I use the semiochemical and the people around me don't?
They will go to the others, or they will come to you.
Yes. Feel free to don't let only him if you want to. The insects from close by fields can immigrate. And so you can reduce the population maybe in your field, but not in the close by. So it's important that everybody uses it. Otherwise, it doesn't make sense. It's efficient as more it is used. Have you ever been here? I recommend you to go. It is on the northeast of Italy, it's called Trentino. This picture is from Piana Rotariana, which is a plain in the Adige Valley River, the Adige River Valley. And here you see mostly vineyards, but there are also apple yards. And in this region, in specific valleys where they produce apple, they were able to limit this pest, the cornel moth, Pseudomonella, by adopting semiochemical-based control strategies, using its main pheromone. And the province of Trento recommended everybody producing apples to use this strategy in order to reduce the impact of the pest on the whole valley. And it worked. They stopped using pesticides in the course of the 90s. They don't use them anymore. They use only semiochemicals to control this pest. This pest is the most classical romantic maggot of apple. You know, since when we were kids, so we know that apple have a warming style. It's this one. Even if there is also the apple maggot, which is Rhagoletis pulmonella, it's a fruit fly from the family. It's not a Lepidopterus pest. But this one is the most common, which is a Lepidoptera, and the pest fruits, biodepositing, and the insect chew the larvae when it's hatched, it chew the epicarp, and then it penetrates in the mesocarp until the center of the apple. And it releases its species, the larval flask, which develop molds and other kind of yeast and various microbes that make the fruits unedible. That is a big issue. It has been a big issue until it was used the semiochemical based compost. The most common insecticides adopted for this pest were organophosphates that you may know that are very polluting, and so it's better to integrate the management of this pest. This is the diffusion. It is diffused in the Mediterranean basin and not everywhere. There are in South Africa, the Checkers, in Australia and in New Zealand, and also in South America and North America. So the biggest producer. Unfortunately, I was not able to get more updated estimation of the cost of these pests, which is very limited, of course. $609 million per China is nothing, and $500,000 for the United States is nothing. I mean, it's the price of a plant with a reasonable size, but still, it is very unpredictable. And despite the cost, the overall cost may be limited, it may ruin the economy or the plans of a business if it's pests with a high infestation. In any case, this is the situation. Every country producing alcohol may have issue with this pest. And as I was said, in Trentino, they are controlling the infestation by using mating disruption of 7,500 hectares out of 9,500, which is the average, which is the total amount of hectares present in the province for the apple production. And the dispensers look like this. There are even other kind of dispenser anyway. The air is permeated, and this is the way it works. This is the compound which was used up to now, is the pheromone called codamol. Codamol is EE810 dodecadene-1-ol, is a long fatty acid hydrocarbon. And so it was first identified by gas chromatography in the course of the 70s and doing electroantenna recording on insects. In few words, they have extracted the female glands, pheromone glands, and they estimated the production of 6.5 nanogram per hour of this compound. And actually, there are still no evidences demonstrating the existence of another compound that is more active than codamol, and it is produced by the pheromone glands of these females, but it's not the only one. There are also some variants like codamol acetate, which is an acetate variant, and dodecadienyl, which is an aldehyde. And there are also geometric isomers at these two in saturation. It can be ZZ, EZ, or ZE, and not only EE, but at minor quantities. In any case, this is the most attractive. None of them is attractive on males, and at the same time, they have to be more or less integrated at specific dosages. The existence of one isomer rather than another, or a little increment of one aldehyde rather than an acetate of one of them, make pheromone races into the species. So the insect from various populations around the world of these species can differentiate themselves in terms, like if we talk about races, because they have different small amounts of different production of this particular compound, okay? Of the various compounds of the bouquet. So it is emitted with many other compounds, so let's say, but this is always the prevalent. So without this, there is no attraction. If there is the other one, the acetate, it means I am a female from a different species. For example, the acetate is present in other species of the genus Celia. So, yeah, this is to give you the idea that it is important that there is a particular compound in the main semiochemical to control. So, why do we study semiochemicals? This is a sort of brainstorming in order to understand the reason. If you feel sick because of a pathogen, because you are infected by something, you use an antibiotic drug to fight it, okay? And so imagine that your crops are sick because of a pest. You can use a pesticide as a sort of drug. Pesticides pollute because they may have off-target effects. So they don't pollute. When we say pollute, it's because they bother somebody else. They bother beneficial insects, maybe. They bother the microbial beneficial fauna or symbionts of other plants. They may pollute us because they bother us if we eat such kind of chemicals. And semiochemicals are alternative drugs to pesticide, and they limit pest population. And so, as I said before, if you have less animals pesting your crops, then you make life easier for the other natural competitors present in the environment, which may be parasitoids or the other different kind of predators, including birds, for example. And less pest means more efficient control strategy integrated with the use of semiochemicals. So you don't only allow the environment to compensate the presence of the pest, but you also can help yourself by having more efficient strategies because you have less pests. Okay? Now we go on the chemosensory receptors of this particular horticultural pest that I was talking about, Cilia pomarella. How do they sense? How do they smell? We use the nose, right? They use the antenna. The antenna is the insect nose. If you magnify by scanning electron microscope the antenna, you will see something like this. There are a lot of areas, let's call it, they look like areas, they're not areas. They are an extension of the cuticle of the antenna containing neurons. Into that, there is the sum of the neurons at the base, and the dendrites of the neurons innervate the intracellular, the extracellular matrix, which is present inside the cuticle. If you see, there are some kind of openings here, some pores. And these pores are the pores that communicate with the outside. The odorant compounds that are present in the environment may have a different nature. They may be hydrophobic, antipathic, or hydrophilic. In any case, over there, in the presence of the cuticle, there is always, since the cuticle chemical composition, there is always a sort of hydrophobic environment that can help hosting hydrophobic volatile compounds. So while the hydrophilic can access directly. Okay? And when they access, they bind specific odorant binding protein. I will show you later a slide with more information about that. And they go and activate chemoreceptors. There are different kinds of chemoreceptors. Are you confident with this image, for example? And this is the plasma membrane of the neuron. And these are proteins crossing the plasma membrane, transmembrane proteins. They may have different transmembrane domains, and they have specific binding sites for the compound. These proteins work like ligand-gated cation channels. When the compound binds, they open a pore, allowing passing through cations like calcium, or most of times also monovalent cations with only one positive charge, sodium or potassium. And there are different types. In the next slide, we will go along with the evolution of these particular receptors. The most trendy, let's say, are the odorant receptor, the gustatory receptor, and the ionotropic receptors, IAS. And there are also the TRP channels. You may like spices, right? You may like wasabi, you may like chili pepper. Okay, these are activators of the TRP channels. TRP channels are present in every motile organism. They're also present in yeast and in fungi. And they are responsible for sensing different kinds of semiochemicals. They are related mostly in the trigeminal sensing, but the other ones are appropriate chemoreceptors for odorants and tasters. Like, for example, odorant receptors normally bind volatiles odor present in the environment that are mostly hydrophobic, gustatory receptors of soluble compounds in general. Ionotropic receptors, instead, they bind acids and amines, the typical odors from fermentation or from the dead organism, or that, like, for example, cadaverine, okay, or spermidine, or various type of polyamines that they bind ionotropic receptors. Then there are also
How do we make our cold semiochemicals in an industrial scale?
That is, these semiochemicals can be either produced by fermentation or otherwise by synthesis, made in an industrial scale.
We made something that, for example, in the GC graph exactly located in where that semiochemical is to make it simulated, or is it exactly the same thing?
What do you mean with I didn't understand this question.
We want to make something that looks like that chemical to that insect, or it is exactly the same?
It is exactly the same. It is exactly the same, but later we will see in the coming slides. The target of studying chemoreceptor is also to figure out how the semiochemical interact to make them better. But in general, the ones that are commercialized, for example, the polyamines, will be exactly the same. They come from chemical synthesis. Don't test me the processes because I'm not a chemist. A very complicated process for synthesis. There are PhD students working only on one synthesis to understand how to make it properly. Most of these processes are industrialized. Indeed, if you go along Sigma, Aldrich, Merck, I don't know if you can go along their website and check for compounds, you can purchase them. Of course, they have a certain level of impurities. They are never pure 100%. And this is due to the industrial process of making it, which may cause some molecules present in the background of your final product. So, in any case, various, to continue with the topic of the chemoreceptors, various type of compounds can bind various type of chemoreceptors. In instant, gustatory receptor detect carbon dioxide, for example, are important to detect CO2. And so I will save concept of the TRP channels. It's good to mention them. Also because the TRP channels are mostly involved in sensing temperature and humidity. In insects, for example, there is a wide class of TRPAs. We humans use TRPV. Different type of TRPV, different genes express different TRPV to sense different temperatures, and the TRPM to sense cold instead, TRPV to sense hot. In an insect, they use a TRPA to sense both cold and hot. They have a various range of temperature sense. Then there are also TRPP that sense cold, etc., etc. But let's don't talk about them. Are too complicated. Indeed, I remember when I was introduced to the colleagues from the Swedish University of Agricultural Science, when I started my project, during my PhD, my project was mostly focused on TRP. And they laughed at me. And they said, come on, you're going to make your life crazy if you want to work with these receptors, because they are very complicated. In addition, they also get a lot of divergence within the same expression, make a lot of splicing, and the protein modified itself continuously. So they are very complicated. Let's talk about the most trendy gustatory receptors, ionotropic receptors, and odorant receptors, which are also very interesting in terms of evolution. Okay, the odorant receptor will be the example that I will bring you the more, because we did a lot of research on that. But this is to summarize you how the compounds access to these receptors. So imagine that we have our sensilla, the cuticle with its spores, and there are the neurons inside. There can be 1, 2, 3, 4 neurons, depending on the type of sensilla, depending on the species. They express specific RNA. Are you confident with this aspect of basic molecular biology, cell biology? Okay. So for the DNA, there is the molecule of RNA and RNA that code for the protein. after splicing and rearrangements, when the protein, when this transcript is the name of this with a single strand is mature, it can be read by ribosomes and make proteins. And the proteins reach the plasma membrane going along a complex pathway of membrane targeting. If they are membrane protein, when in phase of synthesis, they are integrated into the plasmic reticulum, and then they pass through the Golgi apparatus. The guy who discovered the Golgi apparatus was Camillo Golgi, to whom is dedicated also our department. And it was from Lombardy, from the province of Brescia. There is my guy. And so these protein form cation channels. And in insects, when we talk about OR, odorant receptor, which are very easy, these odorant receptor form cation channels by forming tetramers, so complex structures made of 4 subunits, which need to include ORCO, which is olfactory co-receptor, olfactory receptor co-receptor, ORC. Olfactory receptor co-receptor is, let's say, standard, but it's very conserved among every insect. So, of course, there are some modifications, there are some mutations among the various insects, but it is a very primitive odorant receptor that remained in insects. When insects evolved to sense compounds out of water from lands, they also evolved other subunits to combine with it to form these cation channels, which are called ORX. But ORX, usually this X is a number. When we study odorant receptor of a species, we find various number of odorant receptors that we call with various names, OR22A, OR6B, etc., etc. And so they form these cation channels. These are the odors, different odors of environment. The odors that they say that they find their way by binding the plasma, the cuticle in these areas that are particularly hydrophobic or directly are in contact with the acidic environment of the extracellular matrix, and then they are cooked by binding proteins. Binding proteins are soluble proteins that are present into the extracellular matrix and are capable to make these odors soluble because they bind them and they carry them to the odorant receptor cation channel. Okay? This process opens a pore for calcium, and there is the signal transduction of calcium along the whole neurons. And this generates the action potential to the antennal lobe. If you have any question about that, I'm happy to reply, because it may not be very immediate, this process, especially in terms of direction of this action potential to the antennal lobe, which is part of the brain of the insect, taking this information from the various chemosensory neurons that are present on the antenna. Okay? How does it look? Great, you like it? You guys, what do you think? Okay. So this is how it works, and we are studying this. This is what we want to study, because we want to study how these bind this, how the odorant receptor bind ligands. The odorant receptor evolved along protostomes. This is a small lesson of biology. What is the difference between protostomes and deuterostomes? We are chordates, we are deuterostomes. That the main difference comes from the embryonic development between these two different superphyla of organisms. In deuterostomes, in phase of gastrula, in phase of embryonal development of gastrula, there is the formation of a primordial gap, the archenteron. And the blastopore is the first hole of this archenteron to communicate with the external environment. Well, in phase of development of the organism, of the deuterostome, for example, of us, this blastopore becomes the alus. Instead, for protostomes, like insects, it's the opposite, it's the other way around. This blastopore becomes the mouth. And this makes the main difference between us and other organisms belonging to this superphylum of Protostomia. In Protostomia, there are insects and all the various arthropods, including centipedes, including crustaceans, including arachnids, for example, and mites, etc., etc. But there are also other important, interesting phyla, like, for example, mollusks, okay? Gastropods, bivalves, cephalopods. Otherwise, there are nematodes that you may know very well because some of them are important pests for plants. Otherwise, annelids like lumbricus or dendrobenas, so the typical worms of the soil. And these are very nice. Onychophora, the velvet worms. I don't know if you've ever heard about them. They are typical from Amazonia. They are very strange. They look like snails, but they are centipedes. It's a particular. Otherwise, the tardigrades. Have you ever heard about the tardigrades? The water bears. If you go in the field and you get some, you will do it for sure. No, you will not. You will go in the field, you will get some moss, okay? And you watch under the microscope. You can see these strange bears inside that have a very funny face. They are in between. They are in between nematodes and arthropods. Okay, they are very cute. And also the platymids. The platymids, you know about them, are very fascinating organisms. They don't need to reproduce, they just split and they form two organisms from one. Otherwise, they are like some intestine parasites, for example, of animals. They are in the phylum Platymids. Okay, all of these are the protostomes. Okay, protostome. We are chordates, we are deuterostomes like Echinoderma, like the sea stars. Also, these basal metazoans are invertebrates. That's why I say that the word invertebrate is not correct. Because invertebrates is everything except this. I don't think it's scientific correct. Because invertebrates can be echinoderms, can be also basal metazoans and protostomes. Let's call the things as they are. Protostomia and containers. Okay? And sorry for this introduction, but okay. So this is to tell you how the evolution of this chemoreceptor went. Because in some basal protozoans, there were gustatory receptors, which were these proteins with 7 transmembrane domain, like these ones, that were able to sense mostly water-soluble compounds, and they were lost in deuterostomes, but they were maintained in protostomes. And then came ionotropic receptors in protostomes that are related with the ionotropic glutamate receptor, which is a class of receptors that we use in our synapses to give the signal to glutamates. Well, these evolved to sense the environment instead, ionotropic receptor. And then the insect developed the podal receptor. So the insect and the arthropods in general, they developed different subunits of ionotropic receptors as well, and also they developed some primordial ORs, odorant receptor, primitive ORs, among which there were the odorant co-receptor, and then in Neopterans, which are the insects that we most of time we encountered in our life, in Neoptera, we find odorant receptors of various units, various ones. And these are the receptors that we study. And in the next slides, we will see how to sense semiochemicals. Why study chemoreceptors? Like pesticides, semiochemicals can be considered like drugs. Okay, before we were talking about pesticides, also the semiochemicals can be considered like drugs. So studying chemoreceptor may unveil novel semiochemicals. So if we know how the chemoreceptors work, we can also find the novel compounds that bind them. not only the ones that are the mostly used in agriculture. We can find some more. And in addition, we can find scaffolds of the activation because a specific chemical, semiochemical, have a specific molecular structure. But this molecular structure can be rearranged by modifying some substituents, for example, and making the molecule stronger to bind the odorant receptor and to interfere more potently or less with the behavior of the pest insect that we want to target. And this is maybe replying to your question from before. So you can make the molecule exactly the same, or you can improve it based on your findings. And we will see how to unveil these findings in the coming slides. And also, findings and chemicals acting on a species is not limited, because maybe it may be sex specific, because the male and the female express different proteins. The male expresses pheromone receptor to sense the female, but the female mostly expresses odorant receptor to sense food sources for oviposition rather than males. Otherwise, different life stage of the animal can be targeted. There can be compounds specific for larvae, compounds specific for first rather than the turning start of the larvae, otherwise for the adult or the latest stage of the adult. So studying chemoreceptor is important to have an idea of the aspect of chemoreceptor of this organism and to find the ligands which are the most functional. And we are talking about reverse chemical ecology. It's a definition of that kind of chemical ecology that is achieved by molecular sciences, by studying chemoreceptors. In general, you identify the assets of the chemoreceptor of your organism, all of the tools that this organism has to interact with the environment and get knowledge, let's say, from the chemicals which are involved in the interaction with other species, okay? And you screen them. You screen them by expressing them in a heterologous system, and in the next slide we see how it works, and making pharmacology out of them. Hopefully that is clear, because now the presentation will dramatically change towards molecular directions. Is there any curiosity, any aspect you want to dig? You need a break? How does it work? If it's okay, I would continue, okay? All right. So we said a lot of nice things up to now, a lot of theory. Let's see in practice, how to say, we do it, how we isolate and study these receptors. This is the insects, okay? From the insects, you take the organs where the receptors are expressed, the antenna. Antenna is the organ where the receptor is expressed. You need a lot of things, okay? A lot of males or a lot of females, okay? That have to be divided, of course, because they have different parts. Unless you want to unveil the overall aspect of the animal. It depends on your protocol. Let's say that we take only males, not because I like males more than female, it's the opposite, but because the males maybe express pheromone receptors, for example, that sense the odor of female. RNA extraction, you extract the RNA with specific kits that are designed to undertake next generation sequencing in Illumina. You may have heard about these techniques. There are even more advanced techniques now. Illumina is the more trendy and is also very potent. And there are kits to retrotranscribe DNA to cDNA. You may have heard about this method. And then this DNA can be fragmented by these kits that are appropriate to proceed with the Illumina sequencing. The Illumina sequencing is a very technologically advanced approach that allows you to cover the majority of your samples in terms of sequencing. And this is what comes out. In general, when we see something like this, we see a scaffold. So a big piece of sequence on which there are several reads, they are called reads, they are short fragments coming out from the Illumina that map this is all terminology of this method. They will map into specific points of these, to specific loci of these scaffolds, but they overlap also each other. So you have the same information by multiple reads, by multiple small fragments from a loop. And with this, you can identify the sequence, for example, of a specific transcript. And this is what comes out from Illumina. You need computers to do that, and a lot of memory. You need advanced softwares, but also skills and advanced analysis, bioinformatic analysis that come out from years of experiences. Not what to do, but this is still theory. There are several softwares that are capable to do so. So some reconstruct the expressed gene transcripts in the target sequence. Then you need databases also to figure out which transcript you got. So there are databases coming from previous research, previous Illumina sequences, previous annotation from others in the course of the history, where you can compare your finding and check what it is. Okay, and you can say this is most likely, I don't know, deodorant receptor 3. Deodorant receptor 3 is renowned in this species to do this. Okay, and then there are other complicated software in order to polish your data to be able to use them after. If there is any question on that, please don't hesitate. Well, when you have this information, you know which is the sequence, for example, of an odorant receptor, but not only of one. You started from your insect, you have all the repertoire of the receptors. You don't have only odorant receptors, you have also ionotropic receptor, olfactory receptor, odorant binding protein. You have proteins involved into specific cellular processes, so you can classify all this information coming out from the transcriptome into what is their functionality, biologically speaking, okay? And then you will focus on what you are looking for. You are looking for receptor because you want to study what they bind, you focus on odorant receptors, for example. And so, you extract again the RNA, but with a different tip. You need this RNA extraction to produce total RNA on which you can work. You convert the total RNA to cDNA, and you have cDNA sequences. And since it comes from mature RNA, you don't have introns. You have only exons. You are confident with this concept, with the concept of introns and exons, okay? And without the introns, these exons that are ready to go can be directly used to make a protein. So I can amplify a specific gene and express it somewhere else to study. And this is what you do. You design primers with the start and the stop codon. You amplify your origin, you amplify your ortho, and you can use them for heterologous expression. So you have amplicons that you clone into a specific cloning vector, and in the next slide we will see. This is why you do such a complicated transcriptomic analysis, because first you have to see what it is inside there, and then you can amplify it. Is it clear? Okay. This is the Drosophila melanogaster. That is a very interesting model organism that we use to express genes of the pests. We know Drosophila melanogaster since the early 20th century, so more than 100 years. This organism is very easy, it's very nice, it is my favorite animal. And it has 4 different chromosomes in its karyotype. The chromosome first is the sex chromosome. We can have X or Y. And there are the chromosome 2 and 3, which are divided in 2N and 2R, depending on the arm of the chromosome we're talking about, 3N and 3R. And then there is a 4th chromosome, which is tiny. But why are we using Drosophila melanogaster? Because this organism takes 9 days from egg to adult. It's very easy to rear, and you can store different lines of this organism into an incubator, and it's easily feed with a very cheap food. You can make an artificial diet containing everything it needs to survive, and makes all the cycle into these tubes. And it's very easy to access, but especially since it is known since a long time, so important findings in genetics and neurobiology and medicine come from this animal. Consider that the distances among genes is measured in a centimorgan, and Morgan was using Drosophila to study genetics. And they are generated along the years different strain of Drosophila melanogaster, in which, in different position on the various chromosomes, there is this cassette. They have inserted this cassette. This cassette contains this region ATTP that can be used for integration of DNA coming from a different plasmid. Are you confident with the phagus and bacteriophagous viruses that attack? Okay, so these viruses integrate their DNA because of ATTP binding sites that are capable to recombine with the specific regions into the Escherichia coli, into the bacteria that are called ATTP. Okay, these recombinable regions represent an important technology that are currently used in molecular biology to integrate and exchange DNA among organisms, for example. This is exactly how it works. So they have generated different Drosophila strains with this cassette positioned into different loci in the whole karyotype, from the chromosome X to the chromosome 4. Okay? So, imagine that this is the gene that we have cloned. Before we've done the Illumina, we have identified another receptor. We have amplified the gene. We have put it into a cloning vector that is commonly PCR8 from in vitro gene. Well, there is a plasmid that is called BOIS that can be used to recombine it inside. You can use restriction enzyme digestion, otherwise you can recombine it and put it inside. And the result, ATTP, okay? You inject the Drosophila of a specific Drosophila strain that you select upon request. Best gene, California, USA, world universe, okay? Over there is a business, best gene, that does so. And all of these Drosophilas, you provide them these, and you say, put it into, I don't know, chromosome X. At the position, you choose where to put it. You tell them, and this is what happens. They co-inject the plasmid with a cap mRNA to express the recombinase from the phagus that make the job, and you integrate it. Okay? And if you want, you can also remove these, but generally we don't do it, by expressing another recombinase that is called LOXP CRE. But let's don't go too much into the details. To make a long story short, you can bring your transgene into Drosophila. Okay? This is how this transgene expression system is designed. It's called UAS GAL4 or GAL4 UAS. There is another line of Drosophila on which you can put the GAL4 transcription factor under the regulation of a specific promoter. The promoter allows the expression of the GAL4 into a specific cell. For example, a specific class of neurons. Some neurons that you can easily access by doing recording the electrophysiology. This protein binds the UAS promoter under which you put your particular gene and express it. So you have the expression only where this promoter upstream has to work. Is it clear? Okay. And in order to be able to reach that, in order to be able to do the Cal4 UAS expression that I showed you in the slide before, start from the consideration that this is the result that comes out from your business in California upon request. And to reach that target, you have to reach this final crossing step, going along multiple crossing approaches. And in order to do that, I don't want to go too much into the detail of this crossing. If you want, we can go along it in any case, but it's important that you remember that there are some particular genotypes that associate specific phenotypes of the insect. For example, there are, first of all, you start from virgin insects, from virgin flies that you can easily recognize because they are whiter, they have an extended length, and they have a black dot on the abdomen that is the last larval consumption of food. And their wings are closer. You start from virgin because you don't want them having sperm from another fly that may contaminate the offspring. Okay? What do you do with that? You cross it with some flies that have a particular recognizable phenotypes. For example, there is the BLCYO. These two markers, okay, they go together. These two ballasters, they are called exactly. They go together, the LCYO, and make the hair of the fly short and the wings, I would say, curling. So this is an important thing. It is most selective because these insects cannot fly, but they jump, okay? And so apart from the functionality of the thing, is also very useful because when you see the wings like this, it is yo-yo. When you see the short tail, it is BL. There is another one that when they go together, they make the insect black. And one is BM2, which make some short tails on the anthers, which is a specific extra wing that is not properly formed under the wing. And the TN6D instead have a cluster of hair on the thorax of the insect, or like it would be on the shoulder. And you can easily recognize these features. And going along the crossing, based on how they look like, you know their genetics until you have the final crossing. The final crossing, you cross it with something that is called delta-relo. This insect have a big chromosomic deletion in the 12, which contain a specific gene that is the OR22. So these insects don't express the OR22, but you give them OR22/4. So rather than expressing the odorant receptor, they express the transcription factor that activates your odorant receptor. I know it's complicated. How do you see it? Any question?
We need that final insect to reproduce.
We need
that final insect to reproduce.
No, we need the final insect to study the odorant receptor from our pest. Okay? So this is the overall crossing procedure to bring a specific odorant receptor of the moth, for example, into a specific class of neurons of Drosophila, a class of neurons that you can access by doing electrophysiological recording. For example, there is this class of neurons, AB3A, that we know very well where it is located. They are very easy to access by positioning the insect properly, and I will show you how, where you can insert your electrode and stimulate with the compounds. Okay? You will see it in the next slides. So this is a short summary. There is the moth that can sense the fruit, Drosophila doesn't. But upon this engineerization, Drosophila can sense the fruit. The fruit is just an example. It can be a specific odor you want to study, a pheromone of the female, for instance. So you make the Drosophila transgenic, capable to have the same receptor of the moth. Okay? You position the insect under your microscope like this. You need a gas capillary to keep the antenna in this position. because here there are the odorant receptors, the odorant receptor neurons, the neurons where you have targeted expression of your odorant receptors. You insert an electrode, a tungsten electrode into the eye, like a ground, you know, when you hit the light, you need to make the insect like a switch, but you need to ground your electric system. And then you can go and pick the neurons of the fly, going into the sensilla of the fly, where there are the neurons that express the transgene of the moth. I know it's strange, okay? You see, these are the sensilla, and then you go inside a specific sensilla, and the fly is still alive. And this is what you do. So imagine to be inside, and you provide odor. The odor binds the receptor, and you get this. It's like you may have done your plot. You may have gone to the doctor and measure your heart, and you see the spike is exactly something like this. You see spiking, two different types of spiking, and sorry, nothing because it was big. Okay. Two different kind of spiking because you have two different neurons inside. You have neurons with this amplitude and neurons with a bigger amplitude. Okay. And you can recognize that this is the effect of your OR that you have inserted because you have replaced this neuron with another odorant receptor, and this is instead another neuron that can be used as a reference just to see if you are in the right place by testing some compounds active on it. And you can count the spiking upon giving a compound, and you can do exactly the same at different dosages. You can make pharmacology. The orphanization is when you find a ligand, because you start from the consideration that an odorant receptor is orphan until you don't find the ligands it binds. If you orphanize it, it means you have found the ligands. And like this, from a specific receptor of the moth, you are able to identify ligands active on it and do some pharmacological study, doing dose responses, etc., etc., and estimating the properties of this ligand binding on the receptor. This is the apparatus. There is a glass tube into which there is air pumped and humidified, in which you can insert your a small syringe, let's say, into the tube to provide the stimulus. Here you have your insect. This is the recording probe, and this is the reference probe, the one which goes into the eye. Okay? Everything is into a Faraday cage. You can have also an outlet from a gas chromatograph, and we will see later how it works. In order to make these tungsten electrodes so thin to be able to go and to be inserted into the neurons, you can use NOxy reduction using a specific voltage on a solution of potassium hydroxide. It reacts with the tungsten, and then moving to and fro, you make it sharper and sharper in order to be able to access it. Now, if you choose, you can do it under this combo stereoscope. It's very easy to do. Okay. And this is how it sounds when you make your recording. So this is a very tonic effect. You have a basal spiking, that is how the neuron works, because it continuously has positively charged cations going inside, and then they go outside. It's something that happens very fast. It has a basal spiking, but when you give a ligand, this ligand opens the cation channel, and this spiking becomes more frequent, like this, and it makes that kind of sound. And in this case, in particular, I was performing two experiments with two different odorant receptors from two different moths. This is the moth pesting apple, this is the moth pesting the apple bark. And I found this compound capable to bind both this receptor from this species and this other receptor from this other species. That is ZOA32, and we recently published this article a couple of weeks ago. And this is the nodal OR, I cannot tell you which is it. I submitted the paper on Friday. And this is how it works. Any question? No? This is the difference between a very phasic and a very tonic effect. Before I use the wrong terminology. Phasic means a lot of spiking and activation which lasts very short. Tonic is when it goes longer and takes long time to be somehow recalibrated. But still, in both cases, these are two types of activations. Okay? And this is how you can implement your studies by performing gas chromatography, coupling gas chromatography. Are you confident with gas chromatography? How it works? Well, there is this image that can be summarized. You can inject a sample that contains multiple compounds at the same time, because you can get this sample from, you will see in the next slide, from a specific matrix, from a fruit, from a plant, from an insect. This sample contains multiple compounds that, when injected into the gas chromatograph, they are brought up to 200 degrees and they are pumped with hydrogen pressure along the column. They go along this column and they separate each other depending on their volatility and their molecular weight and their affinity with the resin present into this column. This column is very long, various meters. And then they go out and half of the sample goes to an ionization detector that can tell you, give you a peak related to the specific sample, to the specific compound, and the other part of the compound, half of the sample, goes out on the transfer line and reaches the antenna where you have placed the insect. Okay? Reach the preparation where you have placed the insect and prepare the antenna with your connection with the neuron. This is the GC-SSR. Okay? How to collect these samples? I start from an apple, polyester over a roasted bag. Charcoal filter, and you pump air inside. The air goes out and goes to a Pora cube filled trap, collection trap. It's a sort of glass tube, which contains this resin, which can bind the compound coming out from the apple that are pumped out, the apple emit compound. You take them, okay? Then this one, you can wash it with hexane into a beaker, and you let the beaker evaporate, and then you can dilute it and inject it properly with using these very thin syringes into the sample injection system of the GC and perform your studies. This is how it looks. You have to calibrate your equipment in order to be able to know at which concentration you have an activation. For example, I was able to calibrate the equipment to a sensitivity of 1 nanogram, which is you can see something like this sort of peak that is very apparent, but it is there, and you see the effect. I was using a wild type fly, and I know that this compound is active on the neuron that is not transgenic, the wild type. If you don't insert the transgene of your fly, of your moth, you have this compound binding this particular neuron. This is exactly the same connection when I was using 10 nanogram of 2-heptanol. Okay? You see that it's active on the other neuron. There are two neurons, and you see two different spikes, a bigger one and a smaller one. Okay? This is active on the small. This is the compound, and this is the frequency plot. It's a plot that you can derive from your software by setting the software in a way that it shows you a plot rather than showing you the spike. And this is what it came out from Apple on a specific odorant receptor of the causing moth. The odorant receptor 22 is typical for female. female express the odorant receptor 22. You see that here is a lighter rather than make this is a sort of indication. The darker it gets, the less is present. The lighter, the more is present. Okay? And in this case, we noticed that it was specific for female. We wanted to study it in alternative to the ones from male, because most likely this will involve the oviposition. Okay? This is the other technology instead that I wanted to introduce you. We have 20 more minutes. These are human embryonic female cells. Have you ever heard about them? Okay. They are commercially available. They come from an apparently healthy fetus aborted under the Dutch law on the 70s. And they were conducting experiments on tissues from this fetus. And they were able to isolate upon infection with adenovirus. And they were able to isolate tissues that had a very controversial origin. It was unclear if the cells from this tissue were epithelial, fibroblastic, or endothelial. So they collected this particular cell line, which had these mixed properties among different types of cells. And then they also transfected it with the SV40T antigen of another virus that makes it possible to regulate the expression of other kind of promoters. So then you can design specific plasmids with SV40 promoters. And then it's commonly used to express proteins. These cells are very versatile, are very comfortable to use, and are very easy to maintain. And this is what we do. We clone our odorant receptors, right, into specific expression vector, not the PYS, another one. Expression vector dedicated for human embryonic female cells. And transfecting the cells, of course, we give an expression vector with the OR and expression vector with the ORCO. You remember why? Because they don't work if they are alone. And then these cells can express these receptors that upon activation with specific compounds of the cation channels. And if you incubate the cells with ion-sensitive dyes, or if you co-express some proteins that work like fluorescent indicators, when you stimulate the cells, there is calcium going inside. These dyes are sensitive to calcium, and the cells bright. They become lighter. I can show you. Here is an example. This is your preparation of your cells. And when you provide your ligand, you see the cells bright. And by measuring this increment of fluorescence, you can measure the activation of your receptor. You simply do like this. There is a software that you can select the region of interest, probably. This cell, this cell, this cell, this one. This is the reference, the background. This is the background, and this is the incremental fluorescence of all the cells that I showed you before. You remove the background with the software, you bring everything on the on the ground, on zero. Then you can calculate the average and the standard deviations. And these increments are different doses. This is the equipment. There is a perfusion that can be adjusted with several tubes to a rapid solution changer, which is a sort of code that you can command with the software. There is a microscope. Here you have your cells. Here you can perform additional experiments like patch clamp recording, but I'm not going to go into this topic. There is a camera below that can get this information of fluorescence variation, exactly the video that I showed you before. This is the perfusion system with the rapid solution, multi-channel rapid solution changer. This is the sort of code that switch. You can, for example, adjust a specific protocol saying to provide 10 seconds at channel 1. Here there are different channels. You see channel 1, channel 2, channel 3. But your stimulus goes out on channel 3. Then you say, stimulate 10 seconds. So it switches to channel 3 on the cells for 10 seconds. And then you say 120 seconds back. So after the stimulation, go back to the standard wash it. And this is how it works. You can do several types of experiments. Here, for example, we were expressing the OR3 of the condomorph, and we were doing a single sensor recording on the transgenic Drosophila. And we were doing the dose responses at different doses for two different compounds that we found active. This is the elaborated graph with the statistical analysis via ANOVA. And we were able to see that to one compound or to another, there were a different specificity. It seems that there is a higher specificity for this compound because at lower doses also there is a higher activation. We demonstrated the same using the cells. Using the cell system, we were able to see by doing dose response and calculating the half potency specificity. You know what it is? Is the concentration that is capable to give half of the maximal effect. The maximal effect is here, the concentration giving half is the half potency. And this important is an indication of potency of this particular receptor to a particular ligand. For this particular ligand, it was 553, for this other one, 1082. Okay? It means that it's more specific for this because lower concentration gives half the maximal effect. Okay? And this is what you can do. You can do more complicated experiments like testing inhibitors. For example, aminolite compounds are inhibitory for these receptors of the co-receptor. This is an example expressing only the co-receptor, otherwise expressing the co-receptor with OR3, the co-receptor with OR6, the co-receptor with OR1, with other ORs, so the other receptor of the insect. We tested a compound that is specific for orco because also orco has some ligands. They didn't find them in nature, but they found a chemical ligand active on that. And so we have compared different cell plates with different expressions to the same ligand. These are measuring these effects. And then we provided the inhibitor. We tested no activation, you see. And then we washed the inhibitor back again, and we provided again the compound, and we saw amino activation. So different effects from different cells is already a proof that these receptors are expressed because you see different behaviors of these cells to the same compound if they have a different tolerant receptor. These are even more complicated experiments. These are whole cell and outside-out patch clamp recording. Otherwise, monovalent cation permeability study. So imagine to do what you have seen with the insects before, but on the cell, on the single cell. You don't do it with a tungsten electrode, you do it with a very thin borosilicate pipette. It's a sort of soap made by glass. So you pierce the cell physically, and all the material present into the cell diffuse into the pipette. So you have just a membrane at the end, but the membrane contains the receptors. You give the stimulus, and you give higher and higher concentration, okay? And you see higher effect, okay? And why we did this study? To prove that these receptors work ionotropically, like ligand-gated ion channels, because if they work metabotropically, if they need other proteins to work, this would not be possible. Because all the contents diffuse, disappear from the cell, and so an activation would not result in an effect. You know the difference between metabotropic and ionotropic in receptors in general. Ionotropic are the receptors that you give a ligand, they open a channel, ligand-gated ion channel, and they let the cations passing through the membrane. Metabotropic are the one that you give a ligand, rather than opening a cation channel, they activate a protein, that activate another protein, that activate another protein, that activate another one, that open a channel somewhere else. This is metabotropic. In this case, metabotropic activation cannot happen because all the material into the cell disappears because it diffuses. This is another approach. This is a small piece of membrane that most likely contains one molecule of the receptor. You can take the cell, go away with your pipettes, and the membrane becomes thin until it will flip. And you will take a little piece of membrane that you can stimulate. And see, also, by this experiment, you prove that it is ionotropic, because you have just a small piece of membrane that is still active. This is monovalent cation permeability study. It's a very complicated experiment. I don't want to go on the protocol, but if you want, I can explain. To make a long story short, they provided the stimulus by changing into the buffer the different cation, putting sodium, rubidium, potassium, or cesium, and measuring the effect to all of these and seeing the specificity of the response. And we demonstrated that the specificity is proximal for potassium and sodium that indeed are present into the antenna of the insect. These are very sophisticated studies to study the pharmacology of the receptors, to understand the life, death, and miracles of the receptor, a specific receptor of the moth. It may turn useful to add this information to the study of semiochemicals to know their properties, because if you know the pharmacological properties of the receptor, you can make it work better. You can design the more specific semiochemicals that you can use in agriculture. This is a summary for the lesson that, to make a long story short, you have a pest in crop production. For example, the cotton moth that you want to limit. You have to identify your pest that limit your crop production, and you study its ecology. You can have already an idea about the compounds that are present in the environment that this pest can interact with in the ecological interaction of this pest. And then you isolate the total mRNA from the chemosensory organ of this pest, and you identify, as I showed you after the lesson, the assets of the chemoreceptor, the repertoire of the chemoreceptors of this animal that you can study. Then you isolate the coding sequence of some of them, the ones specific for male. The ones that are more expressed in male most likely are the ones sensing the female odors. The ones that are more expressed in female are most likely the ones sensing the oviposition substrate, for example. You need, of course, to go along publications and previous knowledge to have a better idea. You can heterologously express them ex vivo. Ex vivo means out of their original organism. In this case, using transgenic Drosophila that you study in vivo because you keep the animal alive, or in vitro using human embryonic cells because you use a cell culture. And then you can do the functional characterization, the deorphanization, so you find the semiochemicals active on this receptor, and you study their pharmacology. The pharmacology, for example, are all the slides I showed you before, otherwise doing the dose response, understanding which is the most potent ligand for this particular type of receptor that most likely works better as a semiochemical. And then you can identify novel ligands, for example, novel semiochemicals, or you can confirm the activation to previously found semiochemicals. Otherwise, among various semiochemicals that are already known, you can find the most useful, the most promising, that can be formulated and applied in agriculture. But in order to do that, you need the behavioral studies in the lab, in officina. You do behavioral studies, you test your ligand, and you see at which concentration, despite we know the pharmacology, at which concentration it works better. And after having done experiments in officina, you do trials in agro in the field. These are Latin words that I decided to put because they deserve to be used. In agro, because something is working in the lab. Another thing is to see how these semiochemicals can work in the field, because maybe there are other background odors that do not make it functional. And so what we have seen, for example, in the use of pheromones comes from long experience of field trials and important development in their formulation, but especially in including them into proper dispenser, which were able to release them accordingly to the temperature and the various environmental conditions of the field. And then technology transfer, okay? That it's another universe that many times is the most complicated milestone to reach. Okay? Is there any question? I know that maybe it was very boring.
Technical know how to carry out the technique, how to do the graphs,
Yeah.
what's going on.
Which graphs? We can go along it.
I need to really like sit down and study more.
Yeah. This is an overview. I know that is a very complicated, but this wanted to give you an idea of what is the research behind the identification of novel semiochemicals. That comes from efforts in the field and bibliographic research on the species, but especially on heterologous expression experiments of the various ligands. So there is a lot of molecular science, which is the approach of the reverse chemical ecology.
It was very interesting this kind of seminar because maybe in sustainable agriculture you heard about integrated pest management. We have seen also farmers and so on using some techniques, but you sometimes don't know what is the research behind and how to improve all the chemical mechanism to understand.
There are also other systems of expression. They use xenopus frogs also, the oocytes from a frog. That is another more complicated approaches.
So we can actually integrate this one. What is it? Now, insect.
Analysis techniques.
Yeah, if we compare the sterile insect technique with this particular one using the semiochemicals, I think the result would be more, right?
Yes, exactly. Sterile insect techniques was applied starting from insects that were made sterile by using radiation. It's possible and it works much better design strategies based on CRISPR gene editing to make these insects sterile, but according with the European legislation, they're still considered GMOs. So they're not used in Europe. But integrating, as we say, integrated pest management, also sterile insect technique has a huge application in, together with semiochemicals, together with other agronomical strategies. And together with chemicals, because doesn't mean to don't use chemicals, and the most common chemicals to control the insect.
But this procedure of using chemicals, to some extent, we can't consider it as GMO as well?
No, it's not GMOs, because these compounds are naturally present in the environment and they are compounds. They are not organisms. So they are molecules. So they are odors. And the way they are produced may be some based on GMOs, because they may be generated by some modified yeasts, but it stays industrial. And so you just take the, for example, also insulin for nowadays the diabetes is made by yeast, if I'm not wrong, or by some cell cultures. So it's a way or another to produce compound by fermentation. Otherwise, they are synthetic. They come from chemical synthesis. Victoria?
Wow, then we learn the first thing.